be here to chair the session today. I've known Martin for since the beginning of his PhD, in fact, and it's been an absolute pleasure to follow his progress. Just an avid reader of the calcium literature and has a deep understanding of the systems that he's working on, always producing highly impressive work. And Martin, we're really looking forward to your presentation. So over to you.
Okay, thank you very much, Martin, for this very nice introduction. I am part of the lab of Geert Bultink and Jan Parijs. And for the people that know the work that comes from our lab, they know that we're, as a lab, interested in the BCL2 protein family and how it regulates calcium signaling. However, these BCL2 proteins were not initially discovered to be involved in calcium signaling, but their canonical role is rather the regulation of cell fate. And the different BCL2 protein family members can be functionally divided in pro-apoptotic proteins and anti-apoptotic proteins. For example, there are the pro-apoptotic executioners, BAX and BAC. And when the cell is healthy, these proteins reside in an inactive state inside the cell. However, these BAX and BAC can be activated by pro-apoptotic activators, BIT and BIM. And when this activation occurs, BAX and BAC can oligomerize in the outer mitochondrial membrane, thereby permeabilizing this membrane and releasing pro-apoptotic factors like cytochrome C inside the cytosol. This then results in apoptosome formation, caspase activation, and in the end, apoptotic cell death. Both classes of pro-apoptotic activators and executioners are regulated by the anti-apoptotic BCL2 proteins. So there's BCL2, BCLXL, MCL1, and these proteins directly interact with BAX and BAC to inhibit their function, but they can also sequester the BH3-only activators to prevent the activation of BAX and BAC. Finally, there is a fourth class of BCL2 proteins, which are called the pro-apoptotic sensitizers. And the difference between sensitizers and activators is that these sensitizers cannot directly activate BAX and BAC, but they bind the anti-apoptotic proteins at the place where also the activators bind. And so they replace the activators, which then become free and can activate BAX and BAC, and you get apoptosis. How does this work on a molecular level? Well, all of these BCL2 protein family members have something called BCL2 homology domains. And these BCL2 homology domains are important for interactions with other proteins. For example, the anti-apoptotic proteins, the BH3 domain, the BH1 domain, and the BH2 domain, they form a hydrophobic groove on the surface of these anti-apoptotic proteins. And this hydrophobic groove is needed to interact with the BH3 domain of the pro-apoptotic BH3-only proteins, thereby inhibiting their function. So these BH domains are important for interactions within the BCL2 protein family, but also with other proteins. So keep that in mind. As I said, our lab is primarily interested in how BCL2 proteins affect calcium signaling. As you can see, it's quite a complex thing, and by no means this figure is complete. But what I want to focus on today is the interaction between BCL2 and the IP3 receptor. So it's been more than 15 years now that this interaction has been characterized and also in a functional way. So it was the lab of Clark Distelhorst that found that BCL2 functionally interacts with the IP3 receptor. And through this interaction, BCL2 inhibits IP3 receptor-mediated calcium release. When the binding mechanism was closely scrutinized, it appeared that it was not the hydrophobic groove of BCL2, so not the BH3, BH1, BH2 domain, that are important for BCL2's interaction with the IP3 receptor, but that it is rather the BH4 domain that binds and inhibits the IP3 receptor. In our lab, we put a lot of effort to characterize the BCL2 IP3 receptor interaction somewhat further. And indeed, we found that not only the BH4 domain is important, but also the transmembrane domain of BCL2. And in fact, we believe that this transmembrane domain is important to concentrate the BCL2 at the IP3 receptor, thereby allowing the relatively low affinity BH4 domain to inhibit the IP3 receptor. Most recently, however, we found that IP3 receptors without the BH4 binding domain in their central region can also be affected by BCL2. So there is still some inhibition by BCL2, even if you remove this BH4 domain binding region. And so we found that BCL2 interacts with the ligand binding domain of the IP3 receptor, and we believe that BCL2 competes with IP3 for binding to the IP3 receptor, thereby kind of desensitizing the IP3 receptor towards IP3. And the insight in these binding mechanisms of BCL2 with the IP3 receptor seem to be important in cancer, because BCL2 overexpression is something that has been known for a while now to happen in cancers, but it was mostly related to its canonical function. So with the hydrophobic groove, BCL2 would sequester BIM, which normally would go on to activate BAX and BAC, but because it's sequestered by BCL2, cancer cells can avoid apoptosis. And it's this type of cancer cell that is also sensitive towards BH3 mimetics like venetoclax. So venetoclax can bind the hydrophobic groove, thereby displacing BIM. BIM can then go on to activate BAX and BAC, and the cancer cells die. However, there is an important population of cancer cells that have high BCL2 levels, but that are still not very sensitive towards these BH3 mimetics. So together with the lab of Clark Distelhorst, we thought that in this type of cancer that have high levels of BCL2, but do not respond to BH3 mimetics, that there might be a role for the BCL2 IP3 receptor interaction. And indeed, it seemed that some of these cancer cells rely on this interaction for their survival. So we tried to break this interaction and to use the breakage of this interaction to actually kill cancer cells. So in collaboration with the lab of Clark Distelhorst, we developed a peptide that acts as a decoy to remove BCL2 from the IP3 receptor. And the concept is that we took the domain on the IP3 receptor, the stretch of amino acids that is binding the BH4 domain of BCL2. We fused it to an HIV-derived TAT sequence to make it cell permeable, and to try and use this as a decoy to break up the BCL2 IP3 receptor interaction. Through the years, the peptide has evolved a bit by masking a protease cleavage site to make the peptide more stable. And you will see in this presentation that there are different names for the peptide. So it started out as STAT-IDP, and currently we're working with the name BER2, which stands for BCL2 IP3 receptor disruptor 2. So, but these names are all used in this presentation, but it is about rather the same peptide or about the same concept. And indeed, if we use the peptide on certain types of cancer cells, for example, here in primary chronic lymphocytic leukemia samples, we can see that the peptide indeed induces cell death in these cancer cells. So in our lab, we have put a lot of effort into characterizing what constitutes or what factors constitute sensitivity towards this peptide, towards BER2. And a first observation that we found in diffuse large B-cell lymphoma cell lines, which are rather heterogeneous, is that some cell lines respond very well to the peptide. For example, SUDHL4 cell line responds very well to the peptide. Other cell lines do not respond at all to the peptide, in this case, OCI-LY1. And that the response in terms of cell death towards the peptide is related towards the expression levels of IP3 receptor 2. As you can see here, low IP3 receptor 2 levels can be found in OCI-LY1, which is not at all sensitive, while high levels can be found in SUDHL4, which shows a high level of cell death. So there is a correlation with IP3 receptor 2 levels. Furthermore, it seems that for BER2 to kill cells, you need ongoing IP3 signaling. This IP3 signaling is present in a lot of diffuse large B-cell lymphomas. And we found that if we inhibit in a chemical way phospholipase C, which converts PIP2 to IP3, so if we inhibit phospholipase C, we see that BER2 becomes less effective. So apart from the IP3 receptor 2, you need IP3 signaling. And a third observation that we did is that cell lines that are very sensitive towards BER2 are rather not sensitive to BH3 mimetics, in this case venetoclax, and the other way around. Again, we can see that OCI-LY1, the cell line that did not respond to BER2, has a very low IC50 for venetoclax, while SUDHL4, a very good responder in terms of BER2, has a very high IC50 in case of venetoclax. So the current model for BER2 sensitivity that we're working with is that we need BCL2 at the ER. There seems to be the correlation with IP3 receptor 2. And for BER2 to be active, we need ongoing IP3 signaling. However, there is one question that was still outstanding, and that is how exactly does BER2 kill these cancer cells? Because we know it disrupts the binding between BCL2 and the IP3 receptor, but what happens after? And one thing, or we started to tackle this question by looking at what modality of cell death would be triggered by BER2. So using a fluorescent caspase-dependent probe, we saw that if we added BER2 to our cells, in this case SUDHL4, we see that there is an increase in the amount of caspase-positive cells. Furthermore, if we add ZVAT-FMK, which is a pan-caspase inhibitor, we could see that part of the cells are protected against BER2-induced cell death. So we concluded if we see caspases, we can inhibit the caspases and the cell death is not there. That's probably BER2 is triggering apoptotic cell death. And this apoptotic cell death, as I've explained in the beginning, might be due to the interactions between BCL2 protein family members. And actually, we had some kind of hint towards this kind of mechanism, because if we treated SUDHL4 with sublethal levels of BER2, we could see that there is an upregulation of the BH3-only activator BIM, so that can then activate Baxen-Bach and instigate cell death. Moreover, it seemed that this mechanism was calcium-dependent, because if we add BAPTA simultaneously with the sublethal levels of BER2, we do not see the rise in BIM levels. However, when we used lethal levels of BER2, in this case 10 micromolar, in the same cell line, we did not see any significant upregulation of BIM. And in addition, when we used siRNA to knock down BIM in these cells, we could not see any protection against BER2-induced cell death. If something, the cells are a bit more sensitive towards BER2. So it does not seem that BIM is involved in the killing mechanism of BER2. So we had to take a step back. And for that, we looked at something that was very consistent within all the models which were used to study BER2. And that is that it seems that BER2 elicits a cytosolic calcium signal, and that this is a primary and proximal trigger for BER2-induced cell death. For example, if we look again at the panel of DLBCL cell lines, in OCI-LY1, as you can see here, BER2 does not elicit a cytosolic calcium signal, but in SUDHL4, BER2 can elicit a very high cytosolic calcium signal. It is the same, if you remember, when we inhibited PLC, we could inhibit cell death by BER2, and in the same vein, if we inhibit PLC, we also see a smaller cytosolic calcium signal. So it seems that this calcium signal is quite important in the killing mechanism of BER2. So we know what is happening in the cytosol. So we decided to look at what BER2 does with the mitochondria, and we monitored mitochondrial calcium uptake. And as you can see, after 10 minutes of BER2 treatment in ROD2 stained cells, you get these nice oscillations inside the mitochondria. And when we saw that we saw this mitochondrial calcium uptake, we immediately thought about opening of the mitochondrial permeability transition pore, because high mitochondrial calcium uptake or mitochondrial calcium overload is a known trigger for MPTP opening. So for the people that are not familiar with this concept, this MPTP is a pore in the inner mitochondrial membrane. And when a stimulus triggers MPTP opening, this pore renders the inner mitochondrial membrane permeable for all kinds of solids and water. You get mitochondrial swelling. In the end, you get mitochondrial rupture, release of pro-apoptotic factors, and cell death. And you can monitor this MPTP opening rather easily by using a calcine staining. So the acetoxymethyl ester of calcine is cell permeable, and it stains the cells quite nicely. And if you add cobalt, which is a quencher of calcine, you render this calcine staining mitochondria specific, because cobalt cannot readily enter the mitochondrial matrix. If then you add a stimulus that provokes MPTP opening, or that is able to shuttle cobalt inside the mitochondria, you can see that you lose a lot of the fluorescence. So basically, a decrease in calcium fluorescence means that you get opening of the MPTP. And you can see that on these microscopy pictures. So if we add vehicle, after 10 minutes, the calcium fluorescence is still rather strong. But if we add BER2, you can see that there is a decrease in the calcium fluorescence. So it is quenched by cobalt, thereby meaning that MPTP opening has occurred. And after 10 minutes, we get about a 30% decrease in calcium fluorescence. Okay, so we were also able to correlate the mitochondrial calcium uptake that we saw with this MPTP opening. We stained the cells simultaneously with ROD2 and calcine, and as you can see from the dotted line here, if the cell undergoes MPTP opening, simultaneously you can see that the mitochondrial calcium uptake is increasing. And we tried to, with a very simple model, we tried to model it. And this shows that every instance of MPTP opening in our cells is preceded by a rise in mitochondrial calcium signaling. So we know now mitochondrial calcium signaling, mitochondrial calcium uptake is triggered by BER2, and MPTP opening is triggered by BER2. We have a relationship. But what we wanted to know is it a causal relationship, yes or no? So we used this novel MCU blocker, RU265, to block BER2-induced mitochondrial calcium uptake. And as you can see, it works rather well, this RU265, and it is able to, to substantially decrease the BER2-induced calcium signaling. So when we pre-treated our cells with RU265, we saw that the amount of cells that undergo MPTP opening is rather lower if we pre-treat the cells with RU265. So we have an inhibition of MPTP opening, and at the same time, we also have an inhibition of cell death. So this shows that MPTP opening probably underlies the BER2 cell death mechanism. We were able to repeat these findings as well in another DLBCL cell line. So we also see caspase activation, we see MPTP opening, and if we pre-treat the cells with RU265, we also see an inhibition of cell death. And also interestingly, in chronic lymphocytic leukemia patient samples, we saw the same thing. We saw an inhibition of mitochondrial calcium uptake with RU265 here in green. And we could see an inhibition of BER2-induced cell death. So these things seem to hold up. So in conclusion, our model currently is that BER2 is causing a disruption of the BCL2 IP3 receptor binding. This releases calcium in the cytosol. Part of that calcium is taken up by the mitochondria and triggers MPTP opening, which then goes on to caspase activation and apoptosis. So that was the first part of this talk. The second part is more about a recent finding that we've done. So in the search for the BER2 killing mechanism, we, more by accident than by sheer rationale, we used uabain, which is a sodium-potassium ATPase blocker, together with BER2. And what we found is that uabain is apparently protecting cells from BER2-induced cell death. And by what we knew from the killing mechanism of BER2 at that time, we also checked mitochondrial calcium uptake. And indeed, if we pre-treat the cells with uabain, it seems that, that we have a lower amount of mitochondrial calcium uptake. In the same vein, we also find that MPTP opening is reduced if we pre-treat the cells with uabain. So we thought maybe the driving force is different for mitochondrial calcium uptake. So we checked with TMRM whether this was the case, but pre-treatment with uabain did not severely affect the mitochondrial membrane potential. And maybe it's important to say this, but the effect of uabain, the protective effect, is already there after 30 minutes. So if we, right after treatment with uabain, and if we add BER2 then, there is no protective effect, but after half an hour, there is. So it looks like the effect of uabain acts rather upstream of mitochondrial calcium uptake. So we looked at cytosolic calcium release, and indeed there we also found a difference between pre-treatment of uabain and just BER2 signaling. So it seems that uabain reduces cytosolic calcium release. But from our most recent experiments, it does not seem that uabain affects the ER calcium content if we check the ER calcium content with tapsegargin. So at this point, we're not stuck. We have some ideas, but we would be very grateful if some of you might have some input for this story. The open questions that we identified at this point are twofold. Well, is the effect of uabain, is that an on-target effect on the sodium-potassium ATPase? And secondly, is the effect that uabain is having, is it related to the ion imbalance due to lack of shuttling of sodium and potassium over the plasma membrane? Or is it rather its downstream signaling effect? We are aware that there are some papers that claim that uabain is causing some downstream signaling by binding to the sodium-potassium ATPase, but that's something we would like to check. So if you have any ideas about the best way to check this, I will be very open to discuss this. Okay, then it remains me to acknowledge some people. So of course, I would like to thank Jan and Geert for their support during my PhD. Also the other people from the lab. I have presented some work from ex-LMCS members, notably Christina Ivanova, Tamara van Vlussem, Marc Bitremieu, and Haider Akkel. And I would also like to thank our collaborators, especially Clark Distelhorst, Muniswamy Madesh, and Justin Wilson, because these last two provided the RU265 to us. So if there are any questions, I'm happy to answer them, and thank you for listening.
Thanks very much, Martin. A really interesting presentation. There are some questions coming in as they're being typed, possibly. So loads and loads of questions I could ask you, but just to really to get your thoughts. The OCI-LY cells don't respond with the calcium signal to BIRD2, and they don't die in response to BIRD2, and they have low levels of type 2 IP3 receptors, which is the explanation for why they don't respond to BIRD2. But why don't their other IP3 receptors respond to BIRD2?
Well, what we think is, so IP3 receptor 2 should be the most sensitive towards IP3 signaling. And if I'm not mistaken, these OCI-LY1 cells have a lot of IP3 receptor 3, which is not so, or less sensitive than IP3 receptor 2. So what we think is that the ongoing IP3 signaling in these cells needs to get to a certain threshold that activates the IP3 receptor, and this is easier with IP3 receptor 2 than IP3 receptor 3. But actually, I think if there would be a way to increase the IP3 signaling on the condition that BCL2 binds to the IP3 receptor in these cells, but if you would get your IP3 signaling high enough, also these OCI-LY1 cells would respond.
So have you ever tried to give a sub-threshold concentration of an agonist at the same time as the 2, for example, just to soup up just to stimulate the IP3 production to get you to exactly the threshold you were talking about?
No, I don't think we've ever tried that experiment.
Okay, because that would then answer to a degree because if you used a sub-threshold concentration that by itself gave no effect, but just tickled the IP3 production a little bit, then you'd be then you might be able to answer that. So
yes, indeed, it's a very good suggestion. Thank you.
Okay, so going through the questions that are coming through the chat box, thanks to everybody. So Rajesh came in. You might have answered this one. Not sure if I missed it, but did you show the impact of this peptide on calcium release from the ER upon IP3 stimulation of the cells? So I guess that speaks to. I'm not quite sure where Rajesh was going with that one, but have you done IP3 stimulation before and after the peptide, for example? Is it the same pool? How does it synergize with IP3 stimulation via an agonist, maybe?
I'm not sure whether this has ever been done.
Okay.
No, I don't think so. But what we know is that it's IP3 receptor mediated, so the calcium that is released comes from the ER, and it seems that there is also an important component for extracellular calcium that comes in when we add BIRD2. We do not fully understand why this extracellular component needs to be there, but it seems that if we add EGTA to the outside of the cell and we chelate all the calcium, that BIRD2 is working less well. So it's less able to elicit a calcium signal.
Right. So there's quite a few comments about your recent Warbane data. So I'll read these all out simultaneously. I think quite a few of us were thinking of the same thing. So Dan, my colleague Dan Johnson says, great talk. Do you think the Warbane effect could be because of calcium-sodium microdomains? Colin asked, does Warbane inhibit calcium signals evoked by conventional receptors that evoke IP3 formation? I was also wondering whether it affected IP3 production. Kevin's asked, Warbane inhibits calcium release, ... What are your ideas about that? Since ER stores are apparently stable, is crack calcium entry activated? So do you want to just elaborate on those points about Warbane?
These are all good points. So to answer Colin's question, it is high on the priority list to check with conventional stimulations to see whether indeed we also see lower ER calcium release. Apart from that, we were maybe thinking to check indeed the sensitivity of IP3 receptors towards IP3 in the presence of uabain. We were thinking to do this with caged IP3 and try to make a kind of dose response curve in presence and absence of uabain. As for the sodium calcium microdomains, well, we have been delving a bit into the effect that the mitochondrial the sodium calcium exchanger might have, because one of our first thoughts was, okay, maybe you get a rise in sodium inside the cell, and this triggers the reverse mode of the sodium calcium exchanger, thereby actually pumping calcium outside of the mitochondrial matrix while it brings sodium inside. With an inhibitor, we have had some success, but we're still struggling to knock down the sodium calcium exchanger to see whether it really whether this is an important part of the uabain protection mechanism. Then, yeah, as I said, we're very interested to see whether it also impacts downstream signaling from the sodium potassium ATPase, or whether it's rather the ion imbalance that is the main thing here. Yeah. Okay,
interesting. I was just trying back out of memory. I think some, and maybe I've got this wrong, but I did Anita Apiria show some time ago uabain-induced calcium oscillations via activation of phospholipase C, which she thought was interacting with the sodium potassium ATPase. So that would be a stimulation, whereas you see an inhibition, right?
Yes.
It's been years since I read those things.
And I think in that same work, it's also said that these oscillations, they activate NF-kappa B signaling, which is then protective for the cells. But in my mind, this would take a bit longer. So as I said, we get the effects after half an hour of uabain incubation, and I don't know how fast this NF-kappa B signaling really is. So it would be interesting to check, and that's also high on the list, to use an inhibitor of this signaling and see where that gets us. Yeah. But yeah, I don't know.
Okay, interesting. That's a long list. Okay, so John come in with, oh sorry, Thomas first. Nice talk, Martin. Does BIRD2 cause ER stress? Can you see activation of PERK or IRE1?
We have checked that, and in our hands, it does not cause ER stress. We have checked all three branches of the ER stress machinery, and in our hands, we do not see anything there.
Okay. John has asked, could uabain be inhibiting counter-current flow of monovalent ions through the IP3 receptors or TRIP channels, for example? You can inhibit calcium release, of course, if you take.
That is a good question. I have no idea, actually. Okay. I have no idea. As far as I'm concerned, I have not thought about that, but it's an interesting thought.
So does uabain work in your flux experiments? If you add uabain to,
we haven't tried that either. It's rather, this uabain story is rather recent data, so we're trying to get around to explain what we see, but they are all very nice suggestions, I have to say. So indeed.
Okay, so so Kevin, just going back to mitochondrial calcium uptake, Kevin's asked, why wouldn't the vigorous sodium calcium exchanger protect mitochondria from BIRD2-induced calcium overload? I think the idea being that it's a slow delivery of calcium that seems to build up, but the sodium calcium exchanger could, in theory, deal with that.
Maybe, although we have talked to Israel Zekler, who told us that a very small increase in the concentrations of ions might get you a very high change in activity of the sodium calcium exchanger. Rather, to be honest, we also have checked with a sodium dye to check whether indeed incubation with uabain would give a rise in the sodium content of the cell, but we did not see anything there. So either indeed incubation with uabain is not causing an increase in sodium, which is maybe a bit strange. On the other hand, it could be that we are out of the dynamic range of the dye. I am not an expert yet, let's say, in sodium dyes. So this was kind of a one-off experiment just to check, but it does not seem to give any rise in sodium concentrations.
Hey, Martin, can I just jump in here a second to clarify what I was asking about? This is this is independent of the uabain effect. I mean, you showed mitochondrial calcium overload and caspase activation leading to cell death, but normally, I mean, it doesn't look like you've got a real, vigorous calcium signal impinging on the mitochondria, and normally one thinks that the sodium calcium exchanger is pretty effective at dealing with mitochondrial calcium overload. And so just when you uninhibit the IP3 receptor, it's acting at a basal level, which isn't going to be that high. One would imagine that mitochondria should be, the sodium calcium exchanger would be more than up to the job of protecting mitochondria from overload. Any thoughts?
Okay.
Martin, can you go back to your ROD2 slide? Because some of your mitochondrial calcium increases seem quite dramatic, if that's...
This one?
Yeah. So those are sharp increases in mitochondrial calcium uptake on the right, yeah?
Mm-hmm.
Would that, I mean, if you look at the average activity, Kevin, that's pretty modest, but there's some sharp rises in there. Would that speak to your question?
Well, I'm not sure. It depends on what's being measured there, because ROD2 will also be in the cytoplasm. So I don't know if these are really mitochondrial signals.
Yeah.
Maybe.
So presumably, did you check that with antimycin oligomycin, Martin, to show that this was a purely mitochondrial calcium uptake signal?
No, I don't think we've done that in this experiment. But to be honest, if we look at a single cell level under the microscope, I mean, the staining has a mitochondrial pattern.
Yeah.
So we didn't validate this experimentally, but visually everything seemed to be okay.
Yeah, I mean, the thing is, because it's concentrated in the mitochondria, you can, Kevin's right, that the ROD2, and some people actually in the cardiomyocyte field, for example, use ROD2 to measure cytosolic calcium because it accumulate there. And the thing is, because there's more volume in the rest of the cell than in mitochondria, you can have a lower concentration, but still as many molecules.
Yes.
And that can contribute to the signal. One easy experiment is just to either use FCCP or antimycin oligomycin, wipe out the mitochondrial membrane potential. You don't have the driving force of calcium uptake, and then that would show that that would then speak to Kevin's question. And
yes.
But anyway, that's a simple thing to do. But if it was mitochondrial calcium signal that's being recorded there, Kevin, that would speak to a relatively rapid kind of uptake at certain points, which might overwhelm the sodium calcium exchanger, I guess.
Sure. Yeah.
Cool. Okay. Good question. Good chat. Okay. So Colin's come up with an interesting idea about expressing uabain resistant sodium ATPase. That would be an interesting approach to use.
Okay. That's indeed very interesting. Thank you.
Yeah. That would be a neat thing to try. And then Raoul's come in with, thanks for the talk. I don't know if I missed this point. Are you sure that the apoptosis observed with the peptide is due to blocking of the BCL2-IP3 receptor interaction instead of the peptide sequestering an anti-apoptotic protein, which could lead to apoptosis anyway?
That is a very good question. Well, what we know about the peptide is that it disrupts, well, in biochemical assays at least, it disrupts the binding of BCL2 with the IP3 receptor. And as I've shown, the initial binding site of BH4, BCL2, was in the IP3 receptor, was in the central domain of the IP3 receptor. So it was always thought, okay, it releases BCL2 from this central domain. But then we found that it interacts with the ligand binding domain, and also the BH4 domain is involved there. So maybe it is indeed that BER2 is alleviating the BH4 domain not from the central region of the IP3 receptor, but rather from the ligand binding domain, thereby sensitizing it to IP3. And I've had this discussion with Martin yesterday. But actually, we do not know where the peptide acts. It might inhibit all the places, or it might disrupt all the connections of the BH4 domain that BCL2 has, the known ones, the unknown ones. It is very difficult to know what is happening inside the cell. We can, of course, in fluxes, you can do this adding BER2, but proof that that the IP3 receptor and BCL2 are disrupted does not mean that no other BCL2 complex is disrupted. So in essence, we do not know about that. It could be.
Can I maybe add something to? I think also the question relates whether other or canonical BCL2 complexes can be disrupted. And in that sense, I think with Mart's paper, and also now in this work with knocking out BIM, and I think with BAX-BAC, we clearly see a different profile versus, let's say, when we benchmark against venetoclax. So yeah, that we think is independent of, let's say, the canonical pathways there. And the other thing we did to make sure that there was an on-target, that was in the 2013 paper of Haider-Akkel. When we knock down BCL2, then we clearly see that cells they don't like it. There's a huge amount already of cell death, let's say 40-50%. But then when we add the peptide, we don't really have additional cell death. So that was an important control, that it's some on target, which I don't say cannot disrupt other complexes, let's say, or other interaction sites.
Right. Okay, thank you. Okay, so so that was Raul's question. Dan just made a comment that sodium dyes are notoriously tricky. I've obviously tried them, so if you want to chat to Dan sometime about sodium dyes, I'm sure he'd be up for that. Coffee comes in with great talk. A different inhibitor of the sodium potassium ATPase might be worth trying too, just in case which might tell you something about what Wellbane is doing on target or off target.
Mm-hmm.
And April, do you find cells, cancer or others, in brackets, with high levels of BCL2, normal IP3 receptor levels, but are still not receptive to BER2?
I think we have tried this with liver cells, actually. I think we have done this with liver cells, indeed, that they have rather high, or was it that they rather have rather high levels of IP3 receptor and low level of BCL2? But I think we validated this in liver cells, and on the other hand, normal cell, because that's a bit the thing. These cancer cells are rather stressed. They are for a cell, let's say, in a very unusual conditions. So they are very stressed, and they are, what they would call primed for death. So they're rather at the edge of apoptosis, and they only need this last final push. While normal cells, they should have some compensatory mechanisms that are able to counter the killing mechanism of BER2, or they're not so dependent on this BCL2 IP3 receptor interaction for their survival. Right. And we think that it is because these cancer cells are already on the edge that you can give them the final push, while the other cells are not on the edge. So if you nudge them a bit further towards apoptosis, that does not mean that they will readily undergo apoptosis.
Right. Okay. So Kevin's asked, what does the peptide do to cells that preferentially express and survive due to BCL-XL expression rather than BCL2?
Well, I do not have a ready answer for that, but we have recently published that cells that have an acquired resistance to venetoclax, so this BH3 mimetic, that in our hands, they get an upregulation of BCL-XL. And so together with BCL2, they upregulate BCL-XL, so they exert a dual dependency, both on BCL2 and BCL-XL. And if we add BER2 there, these cells that have the acquired resistance are not more sensitive towards BER2 than their counterparts.
Okay. Okay, so if I was to come back in, does BER2 disruption of BCL2 IP3 receptor interaction induce any kind of conformational change in IP3 receptor similar to IP3 binding?
That is a good question, and I have absolutely no answer to that.
I'm not aware of that.
Yeah, we don't have an answer to that.
Okay. Well, Martin, that's got to the end of the formal questions, so we should probably, at that point, stop the recording. But if people want to continue asking questions, then please activate your microphones now. And Martin's agreed to